Good morning, everyone. First, I'd like to thank the invitation of the organizing committee. My talk is about predicting of plant complex traits via integration of multi-omics data. When we trace back the history of genetics from Lamarck's theory to Munder's law of inheritance to the Human Genome Project and to the discovery of CRISPR-Cas9, one of the key questions is to connect genotype with phenotype. The approaches can be generally classified as forward or reverse genetic approaches. With the increase of human population, decrease of arrival lengths, climate changes, and insect dangers, and so on, the most important application of genetics knowledge is breeding, to produce new biological materials with higher yield, higher resistance, higher utilization rate, higher nutritional values, and so on. And the development history of breeding technologies is in sync with the history of genetics, and one of the most recent approaches that have high promises in breeding is genomic prediction. So genomic prediction is an approach that first learns from a population with genotype and phenotype information, then builds statistic models to connect genotype with phenotype. Next, for a new population with unknown phenotype, you can sequence the seedlings to get their genotype in a relatively short period. So you don't have to wait them to grow up till mature. Then you can apply the models to predict phenotype of this new population. So genomic prediction has shown great application in animal and plant breeding in terms of shortening the breeding cycle and improve the breeding efficiency. In previous genomic prediction studies, generally only DNA information of the genome was used, but we know that the development of complex traits were also affected by epigenetic information, such as in transcripts and DNA methylation. However, how those epigenetic information contribute to trait prediction remains largely unexplored. The release of genome, methylome, and transcriptome data and 6-phenome data of Arabidopsis around 2016 allowed us to investigate this question. Here we applied two commonly used algorithms to build prediction models. They are rigid regression-based linear unbiased prediction and random forest. RRBLUB for short is a multiple linear regression model where y indicates the trait value you are predicting, x indicates genotype variance, beta indicates coefficient of each variant, and epsilon indicates the residue. The process of model construction is essentially the process of fitting the coefficients. The absolute coefficients were used to represent the contribution of variance to the trait prediction, which we called feature importance. Random forest consists of a series of decision trees for each feature. When a threshold of the feature was given, the samples were split into two subsets, and the mean square error of the trait values decreased in the subsets. After a few thresholds, samples were assigned to corresponding subsets with certain average trait values. The final prediction of trait value for each sample was determined based on the voting results of all the decision trees, and the average decrease in mean square error of the trait values when a feature was used represents the feature importance of the variant. Here we show the prediction accuracy of 6 complex traits when 3 different omics data were used. in the above models. Pink color indicate genome-based models, blue color indicate transcriptome-based models, and green color indicates mushroom-based models. Models built using different omics data led to comparable accuracy for different traits. Specifically, genome was based for flowering time and diameter of rosette. Transcriptome was based for calling leaf number, and mushroom had similar accuracy as genome and transcriptome for rosette leaf number. Rosette branch number and stem length had poor prediction accuracy, regardless of omics data used, indicating low heritability of those two traits. When examining the importance of gene-based features for flowering time models, we found that there was no correlation between genome and transcriptome data. If we tag features with importance values above 19th centile as important features, only a few important features were identified by both omics data. The same findings were observed for random forest models and for other pairs of omics data. Those results indicate that although different omics data led to comparable prediction accuracy, the most contributing genes reviewed by different omics data are largely different. We further downloaded 426 benchmark flowering time genes that have been experimentally validated to be involved in regulating flowering time, and found that only 2 of those benchmark genes can be identified as important genes by all 3 omics data. 26 genes can be identified as important by 2 omics data, and 140 genes can be identified as important by only 1 omics data. Those results further suggest that different omics data should be investigated to identify important genes involved in the formation of target traits. Next, we asked whether variants of those known benchmark flowering time genes are sufficient to predict flowering time. So we established predicting models using variants of 426 benchmark genes, and for comparison, we also built models using variants of the same number of top important but non-benchmark genes. We found that when genome and transcriptome data were used, benchmark gene-based models have similar prediction accuracy as models built using variants of all genes. However, when mushroom data were used, benchmark gene-based model had significantly poorer accuracy than all genes-based model, and even poorer than non-benchmark gene-based model. This is potentially because the benchmark genes we have known are generally from analysis based on genome or transcriptome analysis, rather than mushroom-based analysis. And this also suggests that additional genes besides those benchmark genes may be also involved in regulating flowering time. So to identify additional flowering time, we examined the flowering time phenotype of several gene loss-of-function mutants. From 21 important genes, which were not reported to be involved in flowering regulation, we do find single gene or double gene mutants of 9 genes flowered significantly earlier or later than wild-type plants. However, a similar proportion of non-important genes are also showed flowering time phenotype when mutated. This might be because the flowering time phenotype was observed only for Columbia ecotype, whereas the feature importance was measured using over 300 stations, which may be ecotype dependent. To test this, we evaluated the local feature importance by examining the Shapley additive explanation values. When we have no informative knowledge, the predicted flowering time for different sessions would be similar. After an informative knowledge was introduced, say SOC1 gene expression, the predicted flowering time is negatively correlated with SOC1 expression, because SOC1 is an activator of flowering time. So the higher the SOC1 expression, the shorter time to flower. Thus, flowering time differences between before and after SOC1 expression was introduced can be treated as contribution of SOC1 expression to flowering time prediction. This is shape value. So higher SOC1 expression, negative shape values, and vice versa. We found that accessions can be clustered into 8 groups using the shape value of top 20 gene expression in the transcriptome-based model. Here, the red font indicates knowing benchmark flowering time genes for the 3 top genes, SOC1, FT, and FLC, which are the hub genes in the flowering time regulatory network. Their shape values were coupled in clusters 1, 2, 3, and 8, but uncoupled in clusters 4, 5, 6, and 7, indicating that regulating of flowering time may be more complex in different accessions than what has been reported. Finally, we asked whether integrating different omics data would improve the prediction accuracy for flowering time. No improvement was found for RR-blob models, but significantly improvement was found for random forest models when genome and transcriptome or 3 omics data were integrated. This is potentially because random forest models also take advantage of interactions between features, which were not considered in RR-blob models. To assess feature interactions, we examined the shape values again. In this figure, x-axis shows SOC1 expression. Y-axis shows shape value of SOC1 expression in the transcriptome-based models for flowering time. Dots indicate different accessions, and the colors of dots indicate expression of the FT gene. In accessions with low or no FT expression, the shape values scales negatively with SOC1 expression, whereas in accessions with high FT expression, this negative correlation slowed down dramatically. This result indicates that SOC1 expression interacts with FT expression in predicting flowering time, which is in sync with their regulatory interactions. Another example is interaction between the CG type methylation of ZTAO and SOC1 expression. In the model built using all 3 types of omics data of knowing benchmark flowering time genes, we identified over 7,000 interactions, where interactions between transcriptomic and methylomic features are most abundant. When visualizing the feature interaction above 19th percentile, we found that the feature interaction network mimics the flowering time regulatory network, where 3 hub genes are also in the hub positions and had the highest number of interactions. So take-home messages, models built using 3 omics data had comparable performances but identified different states of benchmark genes. Gene contributes to flowering time prediction are accession dependent. By integrating multi-omics data to predict plant complex traits, we identified knowing and additional interactions among features from different omics data. Much appreciated for She's Lab and my lab members and all the grants supporting this project. Thanks for your attention.